Okay, so now we're going to move on to some of the consequences of epigenetic changes to the genome caused by environment. And if we have enough time in this one, we'll also move on to how mitochondrial defects can cause disease and how we can combat that with IVF. So let's get into it. Stressful events can cause heritable epigenetic modifications to the genome by increasing or decreasing methylation to certain genes, or histone tail interference or deacetylation. An experiment was done on mice, where mice babies were separated from their mothers unpredictably, and mothers would be forced to swim in a plexiglass tube of cold water for 5 minutes unpredictably. So, not very nice, but the next 3 generations were then not subject to these unpredictable conditions, so they weren't separated from their mothers or dunked in the cold water, but they were subject to a swim test, where it was recorded how long it took them to give up swimming in a water container with no escape. So it's kind of thought if they are more depressed, they're more likely to give up swimming sooner than happier, healthier mice who will swim longer because they're more optimistic, kind of. So on average, the mice that descended from the intentionally stressed ancestors that we talked about just a moment ago, they gave up quicker than a control set of mice who had not descended from those stressed ancestors. So that's what we would expect. But this depressive effect in the mice with the stressed ancestors was reversed via the use of the antidepressive drug desipramine, with treated mice showing similar times as the control mice who didn't have the stressed ancestors. The mice DNA was then extracted from the mother's and male offspring's sperm, and it was found that there was increased methylation around the MECP2 gene, which is responsible for Rett syndrome. There was increased methylation around CB1, the CB1 gene, which codes for a cannabinoid receptor, and there was decreased methylation for the CRFR2 gene, which produces stress hormone. So if we look to the right, we can see that the increased methylation of the MECP2 and the CB1 gene decreases their gene expression. So you see the characteristics of Rett syndrome caused as a result of that, and also decreased expression of CB1 means fewer cannabinoid receptors are being expressed. So your nervous system, which needs these to pick up on signals from cannabinoids that the body naturally produces to kind of soothe you, if you have fewer of these receptors, you're going to have less of this soothing response to the body's own cannabinoids. So that's going to increase the stress of the mice with these epigenetic problems. And then decreased methylation of the CRFR2 gene, which produces stress hormone, is going to result in increased expression of the gene, so more stress hormones being produced, which is making the mice even more stressed. Just in case you don't know what Rett syndrome is, which is being caused by the decreased expression of MECP2 caused by the increased methylation. It is a neurological disorder that mainly affects females, characterized by repetitive hand movements and impairments in cognitive abilities. I just put that there for kind of reference so you better know what it is. So that is in mice. stressful events changing their phenotypes caused by epigenetics, there are a few human examples to take note of. Notably, a similar phenomena happened in humans in Denmark in an event called the Dutch Hunger, where children conceived during the hunger winter of 1944, caused as a result of World War II, they had their genome sequenced 60 years later, and they were found to have decreased methylation of growth factor 2 gene, which is important to gestation, increased methylation of genes associated with cholesterol transport and aging, and increased methylation of the IL-10 gene linked to schizophrenia. So these are some of the epigenetic consequences of these people suffering from severe famine, where if you look to the photo on the bottom right, people were extremely malnourished, and mothers who were obviously pregnant at the time were extremely malnourished also, meaning the offspring they were carrying didn't have the required amount of nutrients when developing, so it caused these epigenetic consequences. And the top right is just an interesting fact that might help you remember. A famous actress, Audrey Hepburn, actually lived through the Dutch hunger, so if you know who she is, it may make this a bit more relevant. Another example, babies born to mothers pregnant during the September 11th attack in 2001 show much lower cortisol levels than usual. So takeaways from the sad mice that we talked about first, the Dutch hunger and 9/11, environmental influences and stress can cause noticeable phenotypic differences in offspring as a result of epigenetic changes made to gene expression. So the environmental stimuli that these individuals had when they were, that either their ancestors had or they had when they were developing in the womb, had epigenetic changes to their genome that changed their phenotypes. I think I'll do the next part as a separate thing, so that's just a brief overview of how environmental stimuli can change phenotype as a result of epigenetic changes, mainly through methylation that we've talked about here, but deacetylation and histone tail interference can do the same thing.